So yeah, my name is Sebastian. I'm a PhD student working in Anna Herland's lab at KTH University and also the Karolinska Institute. So I would like to present to you today the project where we developed the 3D printed co-culture platform that allowed us to control and also investigate the specific interaction points between different types of neural cells. So the brain is our most important organ, and it's really a highly complex network of different types and different subtypes of cells working and communicating constantly with each other. But if something goes wrong, of course, it has quite severe effects on us. So there is a great interest to really try to understand also on the fundamental cellular level how different types of cells communicate or interact with each other. So we were specifically interested in the two cell types, in the neurons and astrocytes. So the neurons, the cells that are electrically excitable cells, the ones that receive, process, transmit, and store informations. And then we have the astrocytes that, among many other functions, important functions that maintain the brain, they also show to have supporting functions for the neurons and maintenance functions there. Because the human brain has really limited accessibility, a lot of the research really looking at the interaction between these two cell types with human material has been done in vitro. So there's lots of different actually publications, but just a few selected things that has been done really like kind of the conventional approach, where you have 2D cell culture, so you have your layer of astrocytes that you see, and then you see the layer of neurons on top. And you could show that really, okay, we can prevent neural degenerations in the presence of astrocytes. It's been shown that the astrocytes increase the electrical activity of neurons in their presence. Then there's also just one out of many, but the example that you can do that also in 3D cultures with hydrogels has been shown the presence of astrocytes to kind of help form neurites and neural circuit generations. However, what we lack a bit in this specific example, that if you really want to have a kind of on the small scale level control the cell-cell interactions, that's not possible. And that's where we wanted to make use of this concept of neural guidance chips. So you can see on the top right actually this neural guidance chip platform that has been around for quite a few years now. And the concept is quite simple because you have two, on the left and right, you have two different chambers that are connected by 10 micrometer wide neural guidance channels. So they don't allow the cell body to enter, but the neurites can enter, and they actually nicely follow these neural guidance channels. So depending on the shape of your guidance channels, you can also shape your neurites and lead them to wherever you want that they grow. And nowadays, thanks to really the improvement in the last years of high-resolution printing, you can also print in really this micrometer resolution that we need. And as you see in the example on the bottom right, then if you can 3D print a structure, then any arbitrary structure that you want to draw or design, you can also kind of make a 3D printed neural guidance structure. So to give you a broad overview of what I would like to present in this project, we use this concept of the 3D printed structures, neural guidance structures. We wanted to develop a way or an approach that we can control where we have astrocytes, where we have neurons, and how they can connect to each other. And then look at, with some examples, look at, okay, how does that now influence the neurons? So just a few examples of, we could also analyze on the effects of that. So we started with designing and printing our 3D printed structures. You can see we used two-photon polymerization printing with nanoscribe and the commercially available resin IPVisual. What is nice is that we can really print in this micrometer resolution. It's non-autofluorescence, it's transparent, so we can use fluorescent imaging analysis later on. And it's compatible with cell cultures models. So on the top left, you see kind of our design structure. And you see a pillar structure, so we call it pillar, and then on the top you see a pillar cavity. So the idea is, if you look at the SEM picture on the right, but then the left structure we call no ramps, you see from the top kind of these pillars and with the pillar cavities. And the concept is that we want to have neurons growing in these pillar cavities. So they can, through these neural guidance bridges that connect the different pillars, they can connect with each other, so they're not isolated and still build a network, but they're actually physically isolated from the substrate and from everything around it that is not connected to. Whereas then on the far right, the structures labeled with ramps, you see on the side these additional ramps, or also on the indicated on the design drawing. There's an additional ramp structure that connects the pillar cavity with the substrate. So now if you have neurons growing in there, they can actually start growing their neurites, grow down these ramps, and then if we have a co-culture of neurons and astrocytes on the bottom in the substrate, then they can connect to astrocytes. So just to kind of point out, that actually means that we have 3 different co-culture conditions in this structure. So we have on one side, with no ramp structures, neurons that are really physically isolated, but they can still communicate through the, they're in the same media, so there's still signaling possible. And we have, kind of like in the conventional 2D approach, where neurons are just growing on top of astrocytes, in the middle of the substrate or everywhere around. And then we have on the right side the kind of the neurons that can connect. They're first isolated, but then they can actually connect and grow their neurites down the structures to the astrocytes. So we started first by then just seeding neurons first. So our approach was really that we have our printed structure, we have a well around, and then we just seed them, and they land and grow everywhere. So that looks then like that. Here you see kind of a confocal set stack image, protection, and the side view in red beta-3 tubulin. But so they grow, of course, everywhere. But the important part is that if you look at inside these, maybe we can use the, yes. So inside these, the pillar cavities, these are the cells that we really want to focus on. And then if we look at them from the side view, you can really nicely see, so here there's a ramp structure present, and then you can see cells are growing on the ramp and can go down and connect. Whereas here, when we have kind of, we don't have a ramp structure, they're really, the ones inside are really physically isolated. So then the next step is, of course, so now we have the neurons and we can guide them, but the next step is that we also control where the astrocytes are. For that we took actually a relatively simple, just probabilistic approach. So what we did is we used here with fetal human, we work with fetal human primary astrocytes, seeded them at very low density, so that we have a low chance of them actually landing inside our pillars. If they did, we then could see that and could exclude that from our analysis. But then we could let them proliferate until the density that we actually were happy with, so you could choose the day of kind of where you stop it in the astrocyte media. And still you don't have any astrocytes growing on top of the pillars. And the last steps then, we seed our neurons at relatively high density, so that they populate actually everything again, but most importantly the top of the pillar cavities, and then really resulting in that we have on the top of the pillar, we have only monoculture of neurons. On the bottom substrate, then we have a co-culture of neurons and astrocytes. So here we worked with actually these lund human mesencephalic cells that are neural progenitors that we can differentiate into dopaminergic neurons. So, and you can see here, because of this transparency, that's quite nice, and we can use fluorescent-based image analysis, and we had these dopaminergic neurons, we could perform some ICC staining and analyze the protein expressions of the different conditions. So we stained here for tyrosine hydroxylase, that is a dopaminergic neuron marker. You can see in green, and just as an example how that then looked like, so we could count the cells, we could analyze how kind of how strongly expressed TH was per cell, and get the results of that. And what was interesting now, so here you can see co-culture compared to monoculture, but actually with all the different conditions, pillars and also control together. But what you see here is that actually in the co-culture it was downregulated, the TH expression, compared to the monoculture, really only focusing on the neurons. If we separate that, look at the monoculture first on the left, we could, however, see that the neurons growing on the glass substrate inside the pillar with ramps or without ramps here, they all had similar expressed TH. So of course that is a good thing that our printed structure does not have any negative effects on the TH expression. And then we saw similarly actually in the co-culture, we had this downregulation compared to monoculture, but there was no differences between all these three different conditions, which led to the conclusion that the physical contact was not relevant here, but it was really something through signaling through the media that caused this downregulation. Another example that we did for also based on imaging analysis was calcium imaging. So here we actually work with rat neurons and rat astrocytes, but the same concept as before, seeding first astrocytes and then neurons. And calcium imaging is a method where we use a fluorescent dye that is intracellularly taken up. And then when the calcium concentration intracellular increases, it gives a spike of, increases the fluorescence. And if you image that and plot it, you can see kind of this typical graphs as you see here. You can nicely kind of count actually the activity, the calcium activity, and the spike rate over time of the neurons. So we did that again first just with monoculture to show actually, okay, we did not see any difference between the different conditions. So again, it was good that our printed structure did not influence it in any way. Then when we looked at the co-culture, what was very interesting there, we could see that if we had the neurons that were on the substrate directly growing on the astrocyte, there we could really see this increased spike rate, so they were signaling more often. However, if we compared the pillar structures, whether they were physically isolated or also they had the possibility through the ramps to connect with neurons, there was no increase of that. So it looked like, or from that we could conclude that yes, if you really have full kind of body contact from astrocyte with neurons, it has a positive effect on the spike rate. But the connections that we allowed through our printed guided structure, that was not enough. And then the connections that they could do with the neurons to the astrocyte was not enough to see the same positive effect there. So that we could kind of nicely show with these neural-guided 3D printed structures. So to conclude, very briefly, so we used IPVGO and nanoscribe to kind of print our neural-guided 3D printed structure. We developed a relatively simple method that still allows us to control and compartmentalize the different types of cells, where they are and how they connect to each other. We could see that with the LUMES dopaminergic neuron cells, actually TH was downregulated based on signaling through the media. And the calcium imaging for the rat cells, we could see that there was an increase when neurons and astrocytes were in direct physical contact, but the connections that we allowed them to have through the ramp. And that leads us a bit to the, of course, what can you continue with that? I mean, the nice thing is that because we use a 3D printing approach, we're very flexible in the design. So, I mean, just one example, but again, you can, whatever you can draw, you can print, if it makes sense, of course, but so you can very easily add more connections point, change the scales, and look at different conditions as well, and see if that has another or different effect there. It is possible then to later on, we had it with some examples, but of course with any neurons that grow neurites, and you can guide them and grow them on the structures. You can interact maybe with motor neurons and then with different types of muscle cells and look at that interaction. Or lastly, the interesting part, of course, is you can have some toxicology studies or disease modeling, which, of course, both of that's what there's a lot of examples of different cell culture models there where that has been done. But so we can just copy that, but add that to our platform that allows us really to control also the connection points and gives us a bit more insight on the specific results. It can be very interesting to see if you get similar results or you see some differences there. And with that, I would really like to acknowledge all the people that were involved in this project. I would really like to highlight Alessandro Enrico, who works now at the University of Pavia who has done a lot of the printing part and the optimization there, all our funding partners. And yeah, if I'm doing a bit of advertising, but if you're interested, our work just got accepted, or was just published last week actually, so it's very nice timing, and if you want to look up more details, you can just scan the QR code, or just ask me questions now, of course. Thank you very much.
Right. Any questions for Sebastian?
Thank you, Sebastian. I was wondering, you talk about the Shona devices where you have those small microchannels where you can separate the somal side from the axonal side.
Yeah.
You cannot reach the same resolution using the bioprinting, right?
So, bioprinting, I guess not, but with, I mean, we could...
3D printing.
With the 3D printing, yes, we can, because you can print, I mean, okay, with photolithography approach, you can go really even below micrometer. What we can achieve is maybe 1 or 2 micrometer.
Can you still isolate the cell bodies from the axons?
So if you want to do an approach where you have 2 chambers, theoretically that's possible. I think then you can still, if you just do it in 2D, maybe the standard chip is easier because the printing takes time. So if you do really a lot of kind of a big structure, then it's just a question of time. So then I think the conventional approach is maybe more straightforward, but you could definitely achieve that as well with the resolution.
Just another quick question.
Yeah.
Is all the system submerged under the same media?
Yes.
Yeah, okay.
So we actually were first culturing it in astrocyte media, and then we switched to the neural differentiation media and kept it in that. Thank you.
Hi. So I don't know if you mentioned this, but what was the purpose of having the ramp?
So we really wanted to be able to control the direct physical connections through neurites. So we wanted to see if there's a difference whether you are still in the same media but no physical connections, or if you're kind of in direct full body contact, and see if that has an influence or not.
But why a ramp instead of like tunnels or something?
I mean a tunnel. You mean like a closed tunnel? Yeah, I mean that would be possible too. Again, it takes more because you're closed, and then so we do coating, and then you have to make sure that the coating actually goes inside. So if you have the top open, we don't have to worry that there's actually liquid inside, and it saves us printing time.
So none of your cell bodies migrate down the ramp, it's just the processes?
I mean, they could to some extent. We did have an opening at the beginning. Maybe it was not really visible, so we had kind of a smaller opening so that they don't migrate. If a cell, of course, landed directly on top of the channel, then that was there as well. Yeah, so there could have been cells in between. Yeah.
Last thing, does the angle, like the degree that your ramp is at, does that matter?
According to some literature that we looked at, yes. If you make it too steep, they cannot really grow up or down anymore. We saw that 45 degrees seemed to work, and we saw that for the growing down, that was still fine. Whereas, and also for us, it was important that the astrocytes at that angle don't start to crawl up, and that we could see as well. So, I mean, you can play around. I think we have some merge that still works, but yeah, you should not make it too flat or too steep. Yeah.
Cool. Thank you.
Yeah.
Appreciate
Thank
it.
Just a short question about the adhesion coating.
Yeah.
Do you have the cell, the same attachment of the coating on the glass and on your resin?
Yes. Yes, and we actually used matrixel coating. So what we just coated. We coated everything with matrixel. So we have that on the IPV show, the printing racing, and on the substrate as well.
Okay, thank you.
Yeah, thank you.
Let's thank the speaker once again.
Thank you very much.